Hi everyone, this is Reema, your biology educator from VLearning. So as you know that VLearning is an online educational platform where we provide quality education for all. So to get all the benefits from our channel, subscribe to our channel and don't forget to visit our website, that is www.v-learning.in, where you will be getting all the pre-recorded videos, the live classes and animated videos, and also the PDF notes of all these lessons. So let's begin today's session. So we are in the chapter that is Cell, the Unit of Life, which is your chapter number 8, and you have learned various cell organelles under this chapter till now, that is endoplasmic reticulum, Golgi bodies, lysosome, and in our last class you have learned about the mitochondria and the plastids, right? So in today's session we are going to continue this chapter and we are going to see the various other cell organelles, and this is the end of this chapter. Okay, in today's class you will be learning about the nucleus, the chromosomes, the centrioles, the centromere, and also the ribosomes. Okay, the centrosomes, centrioles, and the ribosomes, and this is your lecture number 6. Okay, so today we are going to complete this chapter, that is chapter number 8, which is Cell, the Unit of Life. So this is one of the very interesting chapter which you have been learning from your lower classes also, right? So yes, so nucleus. So first of all, you know that nucleus is the brain of the cell. Okay, like we have a brain, so the cell also has got a brain. It is also called as the brain of the cell. Why it is called as the brain of the cell? Because it contains the genetic materials, that is the DNA. Okay, inside the nucleus all the genetic materials are present. Okay, so what are the genetic materials? So if this is the cell, this is the cell, this is the nucleus. Okay, this is the nucleus of the cell. So this is the cell, let's say, and this is the nucleus of the cell, right? So inside the nucleus, what is present inside the nucleus? The DNA is present, right? Okay, so inside the nucleus the chromosomes are present. Okay, and in the chromosomes the DNAs are present. Okay, the DNAs are present, and in the DNA or in the chromosome the genes are present. So what is present? The chromosome, then DNA, then the genes, right? Gene are present, right, inside the nucleus. So these are all what? These are all the genetic materials. Okay, these are the genetic materials. Now the nucleus was discovered by Robert Brown. Who has discovered nucleus? It was discovered by Robert Brown in the year 1831. So nucleus is also called as the brain of the cell. Remember this one. Okay, now who has discovered this? Robert Brown. Robert Brown has discovered nucleus in the year 1831. In the year 1831, Robert Brown has discovered nucleus, and later the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. Okay, so after that what happened? The nucleus was stained by the dyes. Okay, and they found, that is Fleming is a scientist who has found the chromatin inside the nucleus. Okay, chromatin inside the nucleus. First Robert Brown has found the nucleus. Who has found the nucleus? Robert Brown. Okay, now after that inside the nucleus, okay, yes, okay, inside the nucleus Fleming has found chromatin. What he has found? Chromatin. Chromatin are nothing but the bead-like structures. Okay, chromatin are nothing but the bead-like structures from which the chromosome is being formed. Okay, chromatin are what? Chromatin are bead-like structures which are present inside the nucleus, and from the chromatin chromosome chromosome is being formed. Is that clear? So it was given by Fleming. Okay, Fleming has discovered chromatin inside the nucleus. Now let us learn the structure of nucleus. Now how is the structure of nucleus? Yes, if first of all, see, nucleus consists of, inside the nucleus you will see the various parts. Okay, so what are the various 5 parts of nucleus? Okay, what are the various 5 parts of nucleus? Number 1, nucleus has outer membrane. Nucleus has outer membrane. Number 2, it has inner membrane. It has inner membrane. Then number 3, you will see, yes, outer membrane, inner membrane together, we call them as what? We call them as nuclear envelope. Okay, we call them as nuclear envelope. Outer membrane and inner membrane is together called as nuclear membrane. So there are 2 nuclear membrane. One is outer membrane and another one is inner membrane. Next, nucleus has got nuclear pore. Okay, in nucleus you will see nuclear pore through which the molecules will move inside and outside the cell. And also in the nucleus what you will find is the, yes, is the nucleoplasm. Okay, nucleoplasm, then nucleoplasm, then nuclei. Okay, nucleoli. Okay, then yes, what else? Yes, so in the nucleus you will find nucleoli and chromatin. Okay, and chromatin. Okay, so inside the nucleus you will find these various things. Now how is the structure of the nucleus? Nucleoli and chromatin. So how is the structure of the nucleus? So see, this is a cell. This is a cell. Okay, inside the cell, nucleus is present. Okay, what is present? Nucleus is present. See, this is how the structure of nucleus is. Now nucleus has got nuclear membrane or nuclear envelope. This one is nuclear membrane. Then here you will find nucleoli, and also here you will find the chromatin. Okay, let me use the different color. So see, here you will find nucleoli. Here you will find nucleoli. Okay, this is nucleoli. Okay, Li. Okay, and also what you will find? You will find the chromatin. Okay, these are bead-like structures. Okay, chromatin you will find. Okay, chromatin, the bead-like structures you will find. Chromatin. These are the chromatin fibers. And what you will find? Nucleoplasm. Okay, this area is called as nucleoplasm. Okay, right? That is cytoplast. Okay, cytoplast and the protoplast. That is, cytoplast is the area where the cell organelles are lying, and this is the protoplast, together known as nucleoplasm or the nucleoplast. Okay, next you will see what? You will see these are the pores, right? These are the pores. So nucleus also has got nuclear pore. Nucleus also has got nuclear pore, Right. Now, what is the function of this nuclear pore and what is the function of this nuclear membrane? The nuclear membrane, it provides protection to the nucleus. Now, surrounding the nucleus, near the nucleus, which cell organelle is present? You have learned in the previous class, that is the network-like structure you will find here, right? Near the nucleus, we call it as ER, that is endoplasmic reticulum, right? It is called as endoplasmic reticulum, and opposite to the endoplasmic reticulum, what is present? Sac-like structure. We call this as Golgi body. These are the Golgi body or the Golgi apparatus. So is that clear? This is what? This is nucleus, okay? Inside the nucleus, nucleole is present, nuclear membrane is there, nuclear pore is there, okay? Nucleoplasm is there and chromatin is there. We see all the 5 things inside the nucleus, okay? Now, what is the function of this nuclear pore? Now, this nuclear pore is responsible for what? Nuclear pore is responsible for transportation, okay? Transportation of molecules. So, what it is transferring? Here in the nucleoli, what happens? In the nucleoli, RNA, okay? In the nucleoli, RNA and protein is being produced, okay? In the nucleoli, RNA is being produced where? In the nucleoli, okay? This RNA and the protein, it needs to be transported. So how it is transported? This RNA and protein from the nucleoli, it is transported through the nuclear pore, okay? These are the nuclear pores, right? Through these nuclear pores, through these pores, the RNA and the proteins are being transported. Is that clear? So nuclear envelope, it is providing protection. Right? Yes. So see, this is the structure of the nucleus. So next, what do you mean by this chromatin? And from the chromatin, how the chromosome is being prepared? So you have seen this bead-like structure. These bead-like structures are nothing but these are chromatin. These are what? These bead-like structures are chromatin. Now, how this chromatin is being formed? This chromatin are being formed when they that is with the help of histone protein, okay? This is what? This is histone protein, okay? They tangle over the histone protein. This chromatin fibers, they tangle over the histone proteins, okay? And with the help of which they form this chromatin fibers, they form a structure which is called as what? Which is called as chromosome. Is that clear? So this is the structure of chromosome, which is actually made up of chromatin fibers, okay? These chromosomes are actually made up of what? It is actually made up of chromatin fibers. Is that clear? Now, you know that in the chromosome, they have here, that is centromere. They have centromere, and these are what? These are the chromatid, right? These are the chromatid. These are what? These are chromatid. Is that clear? So this is how from the chromatin fibers with the help of histone protein chromosome are being formed, okay? Now let us study that interphase nucleus when has highly extended and elaborate nucleoprotein fibers called chromatin. Now you understood which one is chromatin. Which one is chromatid? The interphase nucleus, okay? The nucleus of the cell when it is not dividing. What is this interphase nucleus? Interphase nucleus. Interphase nucleus. Interphase nucleus. Now what do you mean by this interphase? Interphase means when the cells are not dividing, okay? Interphase means you will learn it in your next chapter, that is cell cycle and cell division, okay? Interphase means when the cells are only preparing themselves, for division, but not dividing, okay? Here not dividing. So when the cells are not dividing in the interphase nucleus, what happens there? It is having some nucleoprotein fibers. These are called as chromatin, okay? They are the nuclear matrix and one or more spatial bodies called nucleoli, okay? So see here in this diagram, so see the inside the nucleus, you will see nucleoli, right? Nucleoli, isn't it? Yes, you will see nucleoli, right? Then it has got what? Chromatin fibers, okay? So see, yes, nuclear envelope consists of two parallel membranes with a space between called the perinuclear space. Yes, one more thing. See, here you can see this space or not. This space, nuclear membrane has having one space in between, right? This space, the nuclear membrane has one space in between them, right? This space they have in the nuclear membrane, that space is known as what? It is known as perinuclear space, right? That space is known as what? Perinuclear space. Is that clear? So this space is known as the perinuclear space. The space in between the nuclear membrane is known as perinuclear space. Clear? The space in between the nuclear membrane is known as perinuclear space, and yes, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. So what is the function of the nuclear membrane? As I said, that it acts as a barrier, okay? It acts as a barrier. The nuclear membrane, it acts as a barrier inside and outside the cell, okay? The molecules will be moving inside the cell, right? And also outside the cell. So the nuclear membrane, it acts as a barrier. Okay, it acts as a barrier for the movement of molecules inside and outside the cell. Okay, so and then the outer membrane usually remains continuous with the endoplasmic reticulum, also bears ribosome on it. Okay, so here I have said that near the nucleus you will see endoplasmic reticulum, and you have learned that some of the endoplasmic reticulum are smooth and whereas some are rough, right? So endoplasmic reticulum, it can be RER or SER, right? So some will be containing ribosomes, okay? Some will be containing, let me take this red color, some will be containing ribosomes. Some endoplasmic reticulum will be containing ribosomes, okay? So ribosomes. And this endoplasmic reticulum are called as what? Are called as RER, right? Yes. Next, at a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of two membranes, okay? So the pores which you can see, here in the nuclear membrane, these pores are known as nuclear. These pores are what? These pores are known as what? Nuclear pore, right? So here you can see various pores, isn't it? So what are these pores? These pores are known as nuclear pore, right? So these pores are known as the nuclear pores. Now, what is the function of this nuclear pore? So see here, the various RNA and the proteins are being produced in the nucleoli, right? This is the nucleoli. So who is going to transport them? How it is being sent out of the cell, right? It is being sent with the help of nuclear pore, okay? So the nuclear pores are sending it outside the cell. So see, yes. Understood? Okay. Now, these nuclear pores are the passage through the movement of RNA and proteins. I have explained already RNA and proteins. What it moves? RNA and proteins, right? So, it moves what? RNA and proteins, okay? So, molecules that flex in both directions between the nucleus and cytoplasm. Is that clear? So, yes, the nuclear matrix or the nucleoplasm contains nucleolus and chromatid. Okay, now which one is the nuclear matrix? It is the nucleoplasm, okay? The nucleoplasm is the nuclear matrix. What is this nuclear matrix? Okay, matrix is nothing but a place, you can say, okay, where other cell organelles are present, okay. So here what you can see, this is nucleoli, okay. So in the matrix, nucleoli is present, right? So matrix, in the matrix, what you have seen, the matrix consists of what? Nucleolus and chromatin, okay. So it consists of nucleoli or nucleolus, okay, nucleoli or nucleolus, you can say. It consists of nucleolus and the chromatin. This red one is what? This red one is chromatin, right? This looks like this, isn't it? So yes, next, the nucleoli are spherical structures present in the nucleoplasm. So in the nucleoplasm, the nucleoli is present. Nucleoli is present. This nucleoli is spherical. This nucleoli is spherical in structure, okay? This is the structure. It is spherical, as you can see, it is spherical in structure, right? Yes, present where? In the nucleoplasm, okay? This area is known as nucleoplasm. Nucleoplasm. Clear? Now, yes, the content of nucleolus is continuous with the rest of nucleoplasm as it is not a membrane bound, okay, structure. It is a site for ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. So nucleoli is the site for RNA. So nucleoli is the site for RNA where the protein synthesis takes place, okay? It is the site for what? It is the site for RNA where the protein synthesis takes place. Clear? So what is this nucleoli? It is the site for RNA, and you know that from the RNA, protein is being formed, right? So yes, this is the function of nucleoli. What is the function of nucleoli? It is the site for RNA and it is responsible for protein synthesis. Now, chromosome. Now you already know the structure of chromosome, right? So see, chromosome looks like this thin thread-like structure. So what are these chromosomes? These chromosomes are thin thread-like structure. These are the genetic material. It consists of centromere. The centromere is responsible for separating the chromatids. This is chromatid, right? So these two are called as sister chromatid, okay? They contain equal amount of DNA, okay? They contain equal amount of DNA. Yes, this is also chromatid. This is also chromatid. Now, what is this kinetochore? This kinetochore is a fiber which is responsible for cell division, during the process of cell cycle. So these kinetochores are the protein fiber. You will learn in chapter number 10. So these are the protein fibers. These are the protein fibers which are attached to the centromere here. Okay, here Here it is attached. On top of the centromere, the kinetochore fibers are attached. This kinetochore fibers, it helps in the process of cell division. All right. It is useful for the process of cell division. You will learn it in chapter number 10 in detail. So for now, remember, this is the structure of chromosome, and it contains kinetochore fibers, and it has got centromere. Here, this one is a centromere. Okay. Yes, now let us see. The interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. Yes, we have already seen. Okay, so inside the nucleoplasm, chromatin are present. These chromatin are loose, like bead-like structures, right? Loose and bead-like structures, right? But during different stages of cell division, cell shows structured chromosome in place of the nucleus. So when the cell undergoes cell division after the interphase, the chromatin, it turns into chromosome. Okay, so the chromosomes are responsible for cell division. So every chromosome has a primary constriction or centromere on the sides of his disc-shaped structure called kinetochores are present, and centromere holds the two chromatids of this chromosome. So see, this is the same which I have shown, that in the chromosome, in the center, what you find? The centromere, right? So this centromere is what holding the two chromatids, isn't it? These are the two chromatids, and it is being attached with the help of the centromere. Clear? Yes. Now, this centromere splits during the cell division. So, the cell division takes place with the splitting of the centromere. Okay, we will study it in next chapter, that is cell division and cell cycle. So, now what are the different types of chromosomes? So, the different types of chromosomes are metacentric chromosome. Let me take white color. Metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric. Now, depending on the position of the centromere, depending on what? Depending on the position of centromere. Depending on the position of centromere, the chromosomes are divided into 4 types. Okay, metacentric, submetacentric, acrocentric, and the telocentric. Now, what is this metacentric? Metacentric means the centromere lies exactly in the center, that is in the middle. Okay, the centromere is lying in the center. So see, this is the centromere. This is the centromere which is at the middle. As you can see, right? The centromere is lying at the middle of the, that is forming 2 equal arms of the chromosome, isn't it? The centromere lies in the middle, and we call it as what? It is called as metacentric chromosome. Okay, metacentric chromosome. First is metacentric. Second one is what? Submetacentric. Submetacentric means, so here is the chromosome. This one will be little bit longer. Okay, not exactly at the middle. The centromere is not exactly in the middle. So the submetacentric chromosome has a centromere slightly away from the middle of the chromosome, resulting in one short arm and one long arm. So here both the arms of chromosomes are same. Both the arms of chromosomes are same, but in this, the centromere is located little bit far away from the middle. As a result, this arm is short and this arm is longer, right? So this is what? This is submetacentric. Okay, this is submetacentric chromosome. This is submetacentric chromosome. Clear? Yes. Now again 2 more left, right? So in case of acrocentric, the chromosome, that is the centromere, is situated close to its end, forming one extreme short and one very long. In case of acrocentric, what happens is one is forming very short and one is forming very long, okay, very long arm. Okay, this is very short and this is centromere. So this is what takes place in case of acrocentric. What is taking place in case of telocentric? Telocentric, here the centromere will be, okay, exactly at the tip. The centromere will be here. Whereas in the telocentric, the centromere has a terminal that is chromosome has a terminal centromere. Here in the telocentric, the centromere will be at the terminal end, okay? The centromere will be at the terminal end. Is that clear? That is terminal centromere, you can see, okay? Terminal centromere, you will see in the case of what? In the case of telocentric chromosome. So these are the 4 types of chromosome. Remember, this diagrams are also important, okay? So yes, this is the picture from your NCERT. Yeah, now let us move into the next topic, that is ribosome, which you have studied in the previous topics also. So see, this is the structure of ribosome, and you know that in case of prokaryotes there are 70S ribosomes, and in case of eukaryotes there are 80S ribosomes. So in case of prokaryotes there are 70S ribosomes. In case of eukaryotes there are 80S ribosome. What is the meaning of S? What does S means here? S means Sedgwick's unit, okay? S means what? S means Sedgwick's unit. So see, Sedgwick's unit. S means what? S means here, S is equal to Sedgwick's unit, okay? Now, 70S is divided into 50S plus 30S. One is large subunit, and this one is small subunit, okay? Now it is like this, okay? This is large and this one will be small. This one will be 50S, this one will be 30S, and this is responsible for protein synthesis, okay? This is responsible for protein synthesis, that is translation process. In the process of translation, it is required, okay? So see here, ribosomes are granular structure first observed under the microscope by George Palade. So who has first discovered ribosome or observed? It is George Palade. George Palade has first observed ribosome under the microscope. Okay, these are composed of ribonucleic acid, RNA, and protein. So in the ribosome, what is present? RNA plus protein. RNA plus protein is present in the ribosome. Okay. So you have seen, right, this is cell. So this granular structure, what you have seen inside the cytoplasm of the cell, these are what? This granular structure are what? These granular structure are the ribosomes. These granular structures are the ribosomes. Okay. Yes. The eukaryotic ribosomes are 80S, yes, and prokaryotic is 70S. So each ribosome has 2 subunits, as I have seen that each ribosome has 2 subunits. One is large subunit and another one is small subunit. So it has 2 subunits, okay. There are 2 subunits, one is large subunit and another one is small subunit, okay. So in case of 70S, it is divided into 50S and 30S, okay. In case of eukaryotes, the 80S ribosomes are divided into 60S and 40S, okay. 60S, 80S is divided into 60S and 40S. So 60S is the larger subunit and 40S is the smaller subunit, okay. So both 70S and 80S ribosomes are composed of 2 subunits. In both, they have got 2 subunits. Clear? This is all about ribosomes. Now, let us see what is the centriole. Yes. Now, inside the cell, there is a structure. Centriole is present only in case of animals, okay, only in case of animals, and this is responsible for the process of cell division. It helps the chromosomes, to align properly for cell division, okay, in mitosis and meiosis, okay. So see here, this looks like this, okay. So these are the structures. We call it as centrioles. They are present, they are facing opposite direction, okay. They are facing opposite direction in the cell, and which is present in the cytoplasm. Now look into the centriole. If you see the structure of the centriole, and if you take this portion of the centriole, so in the centriole microtubules are present. In the centriole, microtubules are present. Now when you look into the structure of microtubules, they are arranged in the form of triplets, okay. There are nine triplets arrangement. So yes, 4, 5, 6, 7, 8, and one more where to do here, okay, 9. So they are arranged in the form of 3, okay, that is triplets, and they are 9, okay, and they are 9, okay. These are all microtubules. This is basically the structure of microtubules, okay, and in center of them you will find hole. So that's why it is known as 9 plus 0, okay. What is the formula? It is 9 plus 0, because they are arranged in the form of triplets, okay, triplets, clear. And how many are there? That is 9. Is that clear? Inside the centriole, the microtubule structure is like this. The microtubules also helps in cell division. It helps the chromosomes to divide. Clear? Now yes, centrosome is an organelle usually containing 2 cylindrical structure called centrioles, okay. So centrosome, so what you have seen, this structure, it is the structure of the centriole, okay. This is the structure of centriole, okay, centriole. Yes, centriole. This is the structure of centriole, and it is cylindrical. It is cylindrical, okay. It is cylindrical, and the centrosome is the organelle. So it is a organelle which is containing 2 cylindrical structure called centriole, okay. Next, they are surrounded by amorphous pericentriolar material, and both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel, okay, like this. So yes, this are centriosome, that is centrosome, they are lying opposite to each other, and this arrangement, okay, that is 9 plus 0, it is known as cartwheel arrangement, okay, cartwheel arrangement. See, they are made up of 9 evenly spaced peripheral fibrils of tubulin protein. So these are made up of what? These are made up of tubulin protein, okay. So these are made up of tubulin protein. So this centriosome are made up of tubulin protein. Clear? They are made up of tubulin protein. So there are 9, okay. That is, they are made up of how many? 9 evenly spaced peripheral fibrils of tubulin protein. So see. So next, each of the peripheral fibers is a triplet. You have seen, right? They are triplets. So each of the peripheral fibers are triplets, right? Yes, they are triplets. Next, the adjacent triplets are also linked. They are also linked with one another. So see, they are also linked with one another, right? Yes, the formula is 9 plus 0, which is not given in your NCERT book. All right. Yes. So the central part of the proximal region of the centriole is also proteinaceous and called the hub. So this part, okay, the central region is called as the hub. This region is called as the hub. If you see inside this structure, okay, you will find like this structure, okay. They are made up of protein fibers, okay, that is tubulin protein. They are made up of tubulin protein. So it is connected with the tubules of the peripheral triplets by radial spokes made up of protein, okay. Which protein? Tubulin protein, tubulin protein. Now, the centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division. So the centrioles, this is a cell, okay. Here the centrioles are facing the opposite direction. They will be attached to the spindle fibers, okay. Here also it is present in both the side and it is lying perpendicular. It is lying perpendicular. How it is lying? It is lying perpendicular. So these are the spindle fibers, and here it will help the chromosomes, okay. It will help the chromosome to divide, okay. So the centrosomes are attached to the spindle fibers. They are attached to the spindle fibers and they are responsible for the process of cell division. Is that clear? The centrosomes, that is centrosome and the centrioles are very, very much essential for the process of cell division. We'll study in chapter number 10, that is mitosis and meiosis. So yes, this is done. We are done with your chapter number 8. So we have covered all the topics. I hope you have understood what is discussed in today's session. That is the nucleus, the ribosome, the chromosome, the centrosome and the centrioles. So this is all about it. If you like this video, do like it and also share it with your friends. So I'll meet you in your next class where we'll be studying with a new chapter, that is chapter number 10, that is cell division and cell cycle. Till then, take care and bye-bye.